Hello guys, my name is Dr. Varun and you are watching chlorophyll classes. In this is a part 2 of your video where we are studying about the principle of inheritance and variation chapter. In this chapter till now what we have covered that who is the pioneer of this genetics field, that is the George von Mendel. He tell us about certain things about the genetics which we are studying. Till now we are seeing that what are the experiments he went through. So one experiment which he went through we have already seen, that is what monohybrid cross means. He checked out how a single gene inheritance goes. Then the next one comes the dihybrid cross. But before moving up to the dihybrid cross, I have given a one question in the last video. The question was simple. We have even did a similar type of way to do it. Let's just try to see it once again. That is what the question was, that if a true breeding violet pea plant is hybridized or is being crossed with the white true breeding plant, then what would be the F2 generation? Okay, so we have to make whole of the thing. How are we gonna be doing it? So let's take that a violet plant, the capital V. This will be the dominant parent, assuming this is the male, another is the female. So this is what your dominant. True breeding means both would be of same, capital or small. Dominant violet one, whereas this is the recessive parent, which is white one. Now what happened? They will make what? They will make gametes. So these are the parents. This is the gametes. So the gametes would be what? Capital V and the small v. They are crossed. And when the crossing will be done, what will? This will be the first filial generation. So what happened? Capital V, small v. So this is what? It is an hybrid progeny. Means genotypically it is hybrid. Genotypically means it is a heterozygous condition. But genotypically. But when it comes to phenotypically, then it is what? All of them, the color would be what? Violet flower. So this is what? Phenotype. Now let's just go into the next generation, because we have to see that what is happening in F2 generation. So in F2 generation, we're gonna be taking two similar plant. They will make what? Gametes. So there are two different types of gametes that is possible. What are the two types of gametes possible? Capital V, small v, capital V, small v. And then there will be a crossing over. So I told you that you can directly do the crossing over here, or you can go with the Punnett square. We're going to be going with the Punnett square. So when we are doing the Punnett square, we are making a square. One side we are keeping it male. Let's keep it. This is the male. This is the female one. And the another side female one. So the male has what gametes? V and small v. They has what? Capital V, small v. And when we are making it up, this gonna be our Punnett square look like. Now when we are seeing it, this is what? This. All the three because first we have to see what? Phenotype. So phenotypic ratio means what? 3 plant would be what? Violet. Whereas this is the only plant which has what? Recessive trait. So ratio 1 would be what? White. Then comes the genotypic ratio. In genotypic ratio, what will happen? Then we will be seeing that this look 1, no one looks similar to it. So 1 is capital V. Then we have 2 like this genotype, capital and the small. So this will be 2. And we are left with only single that has what? This small one, recessive trait. So 1. So the ratio is 1:2:1 of what? Capital means homozygous dominant, heterozygous dominant, and the homozygous recessive one. So this is what you have to remember about the genotype. Moving on this basis, let's just try to do one numerical of it. Okay, so this is the another question that we're gonna be practicing today. Now in this chapter we will be practicing the questions simultaneously, not just only at the end. So the next question that is coming to us regarding this is, if a farmer hybridize a plant with contrasting character of red and white flower, plants are homozygous, okay, with respect to the color of the flower. Now the question goes on that if the F2 generation on continue selfing yields 800 plants, what will be the number of red flower plant and white flower plant? This is a one type of a question. If you understand this approach, then everything is okay. And the white color flowered plant. Now over here the concept of ratios come into this one. So what are the concepts of ratio? So the ratio concept is something like that. This is the total number of plant that is present. You have to find that how many will be the red and how many will be the white. So this is what they're talking about, the phenotype. Color means that physically approach. So what is the phenotypic ratio? This is what they are taking only one character. One character means what? This is a one gene inheritance. One character or factor. That is what? One gene inheritance. And in one gene inheritance, phenotypic ratio in F2 generation would be what? It will be 3:1. 3 would be what? A dominant trait. And 1 would be what? A recessive trait. So we are like over here in this question paper, it's not given, but let's just assume that the white is the recessive trait and this is the dominant trait. So now I have given you more information. So the red color one would be what? 3x, and this would be what? White would be 1x. So we have to, what we have to do? We have to divide 800 into equal parts so that this and this can form. Now when something like this has come, so what you will do? You will write number of dominant phenotype. This we have to find out. How we will find it out? From this 3:1, that will be 3x upon 4. 4 is what? Total. 3 plus 1, that is 4. So we have what? We have made the 4 parts of the 800 and multiply by 800. If we will do this, we will get the answer. So if you want to make it a formula kind of a thing, so how you will make it? You will write what? The dominant is like this. You write it 3x by 4 for this, multiply by number of F2 generation progeny. So this is the formula. Let's just try to solve this formula. So when we are solving this formula, we are getting what? This and this goes with 1 and 200. This will be 2 into, that is 600. So this would be the value over here. Okay. So 600 would be this one. Okay. And then the comes with what? That is the number of recessive phenotype. If you want to use directly the formula, that is also okay, or you can even do the 800 minus 6 total out of this minus that. That is 1x upon 4, that is 800. That is, again this will cut to 200, that is 200 would be the recessive progeny. So this is the way of solving it. Okay, so remember, this type of question can be asked to you. Now similar thing can come in terms to the genotype also, that how many will have this genotype, how many will have this genotype. Okay, so this question try to keep up in the mind. Moving on to the next thing. What is that? That is the next type of experiment that he did. The next experiment is he did that two different contrasting characters, means two different factors he took, and then he try to see that what is happening at that point of view. So that is what? That is a two gene inheritance. So next experiment, experiment number 2 by the Mendel. Okay, so experiment 2 is what? 2 gene inheritance. He checked that, and 2 gene inheritance, it is what? Dihybrid, because you are taking two, like there will be the hybrid of two different characters within a one plant. So this is what? Dihybrid cross. So again, same thing which we have done in the monohybrid cord. Here again we will become what? Mendel, and we will do this reaction. So when the Mendel took, so he took what? He took two of the characters. This time we are taking what? Seed color and seed shape. Okay, so what he took? The factor he took is seed color and seed shape. Now I have told you that initially we are starting it with what? We are starting with the homozygous true lines, means seed color. One we are looking for a dominant parent, and one would be a recessive parent of both the characters in one plant. Okay, so over here is what? We are assuming this is the male and this is the female plant. Okay, now over here the dominant parent would be what? Seed color. The seed color which is dominant one, it is the yellow one, yellow color, and shape is what? Round shape. This is the dominant character. What is the recessive one? Green color and wrinkled shape. So I am making the one. This is the wrinkled shaped one. Okay, whereas the dominant one would be what? Like this. Okay, so this is what we are taking up. Now let's just write their genotype, because since they are true line, how they will look like? This. Okay, whereas this will look like what? RR. You can write back or front, but when you are writing it, don't write it like this. This will be a problematic for you to understand the factors. Okay, so write it like this continuously, one gene then the next gene. Okay, that would be easier for you to remember. Now what will happen? These are the parents. The parents will make what? They will make gametes. So now we have to make what? Gametes of it. Since they have what? Only two types, Y and R. Both the gametes will go together with it over here when they will be forming. So here what will happen? Capital R, small r. Here is, sorry, capital R, capital Y, small r. Now what will happen? It will crossing will occur between it. When the crossing will occur, what will happen? Capital Y, capital R, small r. So this is what? This is our hybrid. So this is what? Our hybrid F1 progeny. So this is what? F1 generation. All will look like this. Always remember, F1 progeny, only one type will come. This is the hybrid ones. And since I have told you what? That this hybrid will have a certain feature. Now here to remember you, that is what? When the Mendel did the experiment, whenever single capital thing is coming also, then it will show the dominant character. So what I told you over here, in the Mendel cross, if an hybrid, sorry, not an, if a hybrid if a hybrid has even one dominant allele, expression will be dominant. Expression, phenotypic expression will be dominant. Remember it. So here capital Y is also coming, capital R is also coming. Capital is for what? It is for the yellow, and capital R is for the round. So the seed will be like this. So F1 hybrid will be like this. Moving on to the next one, that is, then when we do the F2 generation, then what is happening? So for the F2 generation, this F1 generation will act like what? It will act like your parents for the F2 generation. And now they are what? They are all siblings. Okay, so this is what? Siblings are having the inbreeding. So this is what we call it, what? Selfing, right? Now, when they are doing selfing, we have to make the gametes. Here, I will go on to the making the gametes further and let you understand that how we make the gametes. For now, we will understand that there will be 4 different types of gametes present. Now, here comes that how I know that 4 types of gametes present. I will tell you after this whole thing is done. So 4 different types of gametes which your teacher knows that, but how you also knows that we will learn it in the next topic after this. That is what there will be. Now we have to make the F2 generation. The F2 generation will have what? It will have we will gonna be making what? A Punnett square. So let's just make a Punnett square. Okay. So one side is one parent, another side is other parent. So there are 4 gametes present. So I'm gonna be making a 4 this table. Okay. Then over here also 4. What are the gametes? The one gamete would be like this, capital R, capital Y. Another is capital R, capital Y, small r, then is small y, capital R, and small upon small. Okay, now since the parents having the same genotype, means what are the gamete of one parent? This is the gamete of one parent. This would be the gamete of second parent. So that would be also similar. Remember this fact. Now let's just complete this table quickly. The table would be what? Here it will be capital, capital. You also start making with me, so that more you practice it, better it would be. Okay, then this will be this, capital, small, capital RR, capital, small, capital, small. Here capital, capital, this, capital, small, capital, capital, small, capital, small, capital, capital, small, small, then capital, small, capital, small, capital, small, 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 capital, small, capital, small, 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 capital, capital, then small, small, capital, small, capital, small, 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 capital, small, 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 small. Now let's just try to understand its phenotype. Okay, genotype is secondary. First let's go to the phenotype. When we comes to the phenotype, then I told you that even one capital is coming means a dominant color will come. So I am using a different colors and shade to it. Okay, so first is this capital, capital means one capital coming yellow color, another capital round. So means they will be what? They will be yellow and round. So I'm making it with a one single color. If you go here, this is also yellow because capital Y coming, capital R coming means round color will come. Irrespective of, we are not gonna see the smaller one over here because one capital coming means dominant expression, which I have told you over here. Okay, so this is also there. Here also capital, capital means this will also be a yellow round. Capital, capital, this also will be a yellow round. This is also capital, capital, yellow round. Look here, capital, capital, yellow, round, capital, capital, yellow, round. Let's come over here. This is yellow, but this is, both are smaller. Smaller R is for what? For the recessive character, because I told you that for the recessive to show its effect, both has to be in the smaller version. Okay, so here this is what? Something different. So I am making it with another color, that is yellow and wrinkled one with the red color. Let's see it. This will be what? Yellow and round. So this is again this. Then this is yellow but small means what? Wrinkled. So this is again with the red one, because here also yellow wrinkled condition coming. Then see here, this is yellow and this is round, so it will be round one. See over here, yellow but this is small, that is wrinkled, so this will be again this one color. Okay, different one. Then again coming back to the next, that is this is what? Green. We have never seen yet that the green seeds are coming. Plus this is green and round, so we will mark it with different one. Okay, let's just take a better color. This green and round. This also green plus capital R, round, so green and round. This is what? Green and round, so green and round. But this one is what? Green but wrinkled. This is totally different. So this is what? Totally different one. I am making it with this red color. So if I have to write the phenotype of them, okay, so if I have to write the phenotype, so in the phenotype I have what option? I have an option of round, 1 minute, I have an option of round yellow. How many round yellow present? Let's try to calculate it with the yellow color I have made it. So we have what? We have 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so how many of them are there? 9. So round yellow is 9. Coming back to the next thing, we have what? This red color one. This red color is what? Yellow wrinkled. So this is yellow wrinkled. How many of yellow wrinkled are present? If you see that 1, 2, 3 only. So we have what? This is 3. After that, we have this color, green. How many of, what are they? They are green but round in shape. How many of them are there? If you see 1, 2, 3. So the ratio would be what? 3. And the last one is what? Last one is the recessive trait, pure recessive trait, which has again shown up here also. Same like the monohybrid and dihybrid cross. So this is what? This is the green wrinkled. So this is what? 1. So the ratio is what? 9 ratio 3 ratio 3 ratio 1. Now if you are seeing this one, here something different happened. What something different happened over here? Like if the monohybrid cross is seen, what are the similarities? The similarity is there, here there is what? These things present, which look like what? Which look like their original parent. Original parent means what? There is a wrinkled, means total recessive trait, which is not present in F1 generation, but it is present here again. This is similar like the monohybrid cross. But what is different than the monohybrid cross? The difference than the monohybrid cross, in the phenotypic ratio only we can see what? We can see that this yellow wrinkled and green round means there is what? There is a mix match. How the mix match? If we go back to the monohybrid cross, see over here, in monohybrid cross, what is happening? That your violet and white means only this factor coming, which is the parent, initial ones. There is a nothing called in between thing or something mixing matches occurring. Means when more than one genes we are taking place, then mix matching takes place. How this mix matching takes place? Here the understanding has to be developed. This new recombinants that are formed, because these are what? These are parental phenotype. Parent was also looking like what? Round, this green wrinkled. So this is coming. But nowhere, not even the hybrid, not even the parents, anywhere this thing has been appeared. So we come to know that what over here, that these are the non-parental combination that is being formed. And these non-parental combinations are what? These are the recombinants. So if I have to write it up, that there is what? Here the difference with the monohybrid is what? That non-parental combinations occur. Combination occurs. Now the question is that how it is occurring, sir? Okay, let's just try to understand from the cell chapter that you might have learned in the 11th class. In that chapter, you might have studied what? When the gametes are being formed, gamete formation time, what has happened in the diploid? Since these are what? We are talking about the diploid organisms. The concept becomes little more titurated. Okay, so what is happening when the diploid cells are there? Then what happened? These are your new, okay, let's see that. This is your chromosome. Okay. Now, if you remember, when they are making gametes, gametes formation means what? There will be the process of meiosis that will occur. For the gamete formation, there will be meiosis process. So, during the meiosis, what is happening? These chromosomes, they look something like this. Like this. Chromosomes, like chromatid numbers are like increased. Okay, they are bivalence. Okay, tetravalence. Sorry, bivalency is there. Tetras formation is there. Now, during this time, what is happening? Certain genes are present. For example, these are the one gene. This is another gene that is present. Here, what happened? They get crossed. This thing can come here, and this thing can come here. And this crossing only is occurring, which leads to what? Non-parental combination. So what I said? I said that during crossing over, genes get crossover from one chromatid to another. Now, when this crossing over is occurring, this crossing over is occurring during the prophase 1. Prophase has 5 different stages. Which stage of prophase 1 has crossing over occurring? We have what? Leptotene, zygotene, pachytene, diplotene and digenesis. So during the pachytene, third one, we have what? This thing, crossing over. So pachytene stage of the prophase 1, this is occurring. Okay, so little bit we understand that how these non-parental combination occurs because of crossing over during gamete formation or the meiosis 1. So this is the understanding thing that you have to remember. Okay, so it is linked. Now when it comes to the phenotype, so if you have to write the phenotypes, so many different phenotypes are there. So if you see over here, this is a different phenotype. So one would be what? YYRR. This capital Y, capital R, small r. This is what? Only 2 present over here if you calculate it. So 1:2 something like this will come up. That is your homework. The genotypic ratio of F2 generation is the homework. We will try to see that in the next class again. Okay, so this is the dihybrid cross. And here, if I have to write it in much better way, so these are what? These are the non-parental combination, or we call it a recombination. Okay, recombinance version. And these are the parental combination. So in your exam, there might be terms coming like what? What is the phenotypic ratio? What is the phenotypic ratio of parental and recombinant? The recombinant is what? Non-parental. Don't get confused. So that is what? Your parental is how many? 9 plus 1, that is 10. So 10 are the parental and the recombinant are what? 3, 3, 6. And since 10 ratio 6 can be further divided into lesser, that is what? 5:3. So whatever could be given to you, you have to remember it. Okay, this is one type of question that can be asked to you. Moving up to the next one after this. So the next thing that is that after the two laws, like after the two experiments, Mendel's gave some of the laws. So these are called as postulates of Mendel's or the laws of Mendel's. Okay, So these are what? Laws of inheritance by Mendel. Now in this, the first law that is there, that is the law of dominance. The law of dominance, or you call it also it is called as law of recessiveness. That I will let you know how it is called as. So it is also called as law of recessiveness. Now when we are studying this law, here we have to remember that, let's understand the basic things. Okay, yesterday class also I went through it. Here we are summing it up with more better way. So I told you that the law, if we are taking it as a law of dominant, the law of dominance will be called like what? That it is a law which states that the trade is what? Your factor, okay, which expresses itself in the F1 generation when a cross between a contrasting character is made. Two contrasting character of same factor is made. The one which phenotypically expresses itself in the F1 generation is over and masking the effect of other one is known as a dominant character, and this is the law of dominance. Okay, remember that eye color example I have given, same thing over here. Okay, next thing, if we have to note it down with terms of recessiveness. So law of recessiveness states that when we are we are crossing between a contrasting character, the character which do not showed up in the F1 generation is known as the recessive trait, and this is the law of recessiveness. Okay, let's see that what it's written over here. So over here it is saying that the law states that when two different alleles of a trait are present, means when the two different contrasting characters present for a gene, one allele will mask the expression of other. The one which will mask means which will stop, hide it, means in case of my brown or blue color eyes, the blue color is being masked, hide up, okay, by the brown one. Okay, so the alleles that expressive is called the dominant allele. Okay, means one allele will express it will be the dominant. Same like that when there are two siblings, the one sibling will be the outsmart the other sibling at lot of places. So the one which is outsmarting the other one, so means one is masked. Okay, so this is what, and the one that is masked is called the recessive allele. So one we know the dominant allele, recessive allele. For example, in the Mendel P experiment, the allele for tall was dominant and the allele for dwarf was what? Recessive. So that's how he gone through all of these experiments to come up to the conclusion that these are the dominant one, these are the recessive one. So the ones which we have studied in the last class, that yesterday class, in that what happened, the dominant one that was coming up, that is what? You're a dominant allele. Okay, so all those dominant allele he able to find it out by doing this whole experiment. That one generation he went through it. Okay, so if we have to write it with the example of this tall plant, so how you will write it, represent it better in your board exams would be something like that. You will be taking the parent 1, parent 2, this is a tall parent and this is the small parent. So it is what? It is a homozygous dominant, means tall parent. It is a homozygous recessive parent, recessive, that is dwarf parent. Now what will be the next step? There will be what? Gamete formation. So here it will be what? Gametes. This is what? Your parents. So this is what? Capital T, small t, gametes. Now what will happen? You will simply make it like what? Crossing occur. Now this crossing will be what? Capital T, small t. This is what? Your F1 generation, or you call it hybrid progeny. So phenotypically it is what? Phenotype, it is tall. So tall becomes over here as a dominant allele in this, because it has both the allele, but the ones which is expressing will become what? Dominant allele. So this is how you will be explaining it up. Okay, so law of dominant or law of recessiveness. Moving on to the next one. Okay, something has been missed. Law of recessiveness. Okay, then comes the law of segregation, or it is also called as law of purity of gamete. Okay, so another name is also there, that is what? That is law of purity of gametes. Now what is happening? If you see across, okay, like F2 generation, when the hybrid is there, hybrid is making the gametes, then we said what? Two of the gametes are going away. So when the gametes are being formed, the half of getting a dominant one, other is getting the recessive allele in the hybrid, when the hybrid is acting like a parents and forming the gametes. Now when they are doing it, is it like that this is being contaminating this or this is contaminating this? No, the pureness is still intact. No one is contaminating it. Means when the segregation is occurring, then the pure form of allele is coming up. No different or no contamination occurs. Means in short that if you and your friend is there and one is not very good in studies and you are good in studies, let's take an example. So maybe you can make that person good in studies by giving the attention, by telling and helping him, or else he can make you worst in studies. So the same thing do not happen at this level, at the gamete level. Means when I will segregate your seats, I will put one good student at other place and the bad student on the other place, still the bad remain bad, good remain good only. So no change in the company. Okay, no bad company affects the aura of the other person. So let's see that what they are telling us about it. This law states that during formation of gametes, means sperm or the egg cells, in case of plants it is what? Pollen or the, means male gamete inside present is the pollen grain and the egg cell. The two alleles of the trait separate from each other. That is what I told you, that when the hybrids are being formed, they have what? Two different alleles present. They will segregate out so that each gametes carry only one allele of the trait. This segregation ensure that the offspring inherit one allele from the parents. Okay, and that too in the pure form. Okay, so that is the major thing. Now if you have to make it up with the explain it up, so for example, in Mendel cross of tall plant with dwarf plant, F1 hybrid when forms gametes, then capital T allele or the dominant allele and the small t allele, that is the recessive alleles, segregates free from each other in independent manner without contaminating each other characteristic or trait. So if you have to make it, so this is the F1 generation, this is the progeny, this is F1 generation. Now when they have to do the crossing, what will happen? They will make gametes, so capital T and small t. So here what happened? This is pure tall allele is segregate out and pure dwarf allele segregates out. So that is a important thing. No contamination occurred over here. So this is the second law. The third law is independent assortment. Remember that when we have made the gametes, now the gametes has choice to go with this and this. Means when I was doing this thing, like over here, let's see that over here. So see over here, this thing has an option to go with this or this. This is only we are showing it over here. So this is what independent assortment means. Every allele has an independence or an independent choice to go and join with everything. Means here what a probability case came up, that this would be the probability that it can go with join with this and form this kind of a further next zygote. Okay, so let's just try to understand what they have written it over here. This law states that the inheritance of one trait does not influence the inheritance of another trait. This means that the genes from a different traits are distributed independently of each other during the gamete formation, provided they are located on the different chromosomes. Here, this thing is important to understand. Now, for example, what is happening? For example, this is your chromosome. First example. So what happened if these genes are there? And this is another chromosome, and on this genes are there. So means, for example, if capital T, small t is present over here, and over here capital R, small r present, means this chromosome can go with this also, this chromosome can go with this also. Or another option is what? This chromosome and this chromosome. So means 4 different gamete options are there. So this is what it is saying over here. So if you have to write it up, you will write that when we are in F1 generation, when gamete forms, factors has choice to assort themselves independently. Okay, so this thing you have to remember. So how you can write it? Capital T, small t, capital R, small r. So option is what? Simply we will write it capital this. So this can go with this capital T R, this can go with capital T R, this gamete can form like this, and this gamete can form like this. So this thing is there. So 4 different gamete options will be here. I hope this thing is understood. Go through it and let me know in the comment section about it. Moving on to the next thing after this. So 3 laws were there. We have done it. What were the 3 laws? Law of dominance, also called as law of recessiveness, law of segregation of gamete, which is also called law of purity of gamete, and the third is law of independent assortment. Coming up to the next thing, that is how to find out how many gametes are formed and what all the gametes genotype would be. Okay, so that is an important NEET question. So what is the NEET level question? That is to find the number of gametes, means types of gamete formed and their genotype. So this becomes a important question. Okay, this we have to do it. So let's tackle with the first question. The first question is that how many number of or types of gametes will form. So first is formula for finding types or number of gametes. So how you will do it? Now it depends upon the number of heterozygous genus present. Okay, so what I said, that depends upon number of heterozygous genes present. For example, okay, so let's not like the goes of for example, let's try the formula first. So what would be the formula for it? Formula is 2 raised to power n, where n is number of heterozygous genes or traits present. Let's just see that example wise. For example, in a monohybrid cross, we have to see that when a hybrid is making the gametes, how many it will make, because the pure form will make only one type of gametes. So here only I am writing that pure form means what? Homozygous will always form one type of gamete, because no options are there now, because there is no difference or heterozygosity. But in monohybrid cross, okay, in F1 generation, hybrid has one heterozygous genus, one heterozygous trait, like for example capital T, small t. So since it has one, so let's put it in the formula. This, it has what? One only. So we raised to power 1. That is what? 2. So remember, we used to make gametes what, like that. One gamete will be T, next would be the smaller t. Forms 2 gametes. Now you will be like, sir, this never happening. Let's see, check more. Okay. So now in dihybrid, there will be what? For example, capital R and 1 we have done it. And over here we obtained the hybrid. Capital R, small, this. So here is what? This is 1, this is 2. So we have what? 2 heterozygous trait. One is for what? One is for the shape of the seed, other is for the color of the seed. So this is what? 2 heterozygous characters, 2 heterozygous traits. Let's put it in the formula. 2 raised to power n, that is n value is what? 2. So 2 raised to power 2, that is 4. Means 2 multiplied by 2 is equals to 4. Now let's see that whether they will make 4 types of gametes. Yes, they will make. We remember that when we are making it, the gametes were like what? That is one gamete is, okay, let's just see this. The one is this capital R has option to go with this, and this means 2 option. This small has 2 option, this 2. That is 4 options. So this would be the way of doing it. Now if there is a trihybrid, till now we haven't made it, so but we will do it. So if it is a trihybrid means like for AA, BB and CC, means 1, 2, 3. So 3 heterozygous traits are there. Let's put it 2 raised to power 3, that is 2 into 2 into 2, that is 8. Let's see that whether there will be 8 or not. We have to confirm it, and this becomes the second question. The second question is what? That to find out how many different genotype of it, because here we can find the genotype because sir has already told it in the last class. Okay, let's just go further into it, how to find it up. Okay, and then we will wind up with this video. Okay, so for this we will be using the Forklin method. Forklin method for what? To find out the all possible genotype of gametes. Very important. So how we will do it? Let's see that the easiest one, then we will go with the further ones. Okay, the easiest one is dihybrid cross. In dihybrid cross, what we have? We have R, capital small. So now what you will do? We will take 1 by 1 things. Okay, what will be the 1 by 1 things? Number 1, we will take this. We will take this one. Then we will go with this one. So what I told that 2 options are there. Okay, so what would be the 2 option over here? The 2 option is, one could be the capital R, other could be the small r. And then this capital R has option what? To go with this and this. Means this thing will further has 2 options. So I will write it up. So the 2 option would be what? One is the capital Y, one is the small y. Same this thing will be here, that this small will has 2 options. One is this, one is this. Okay, So what it will be? It will be capital and the smaller one. So what would be the, after that nothing left. So we will be writing it up together. Then it will be what? Number one would be capital, small, then will be capital and the, sorry, capital first, then the capital small. This is this will be this and this. So these are the four options that is formed. Go back and see that whether this I have wrote or something else I have wrote this one. So whenever I have to find the gametes, I will do this Forklin method. Let's just do one more and then we wind up. Okay, so another one is what? Your tri-hybrid cross. So this is AA, BB and CC. Okay, so let's just make the C little different so that it's not a confusing, like C, smaller, small is this only. Okay, so first option is what? Then I will divide this to, okay, so this will be what? Capital A, small a. Next option is I will divide this to both. So this will be what? Capital B, small b. Here it will be what? Capital B, small b. Then I have option to do with C. So there will be what? Capital C, small c. Here also capital C, small c. There will be what? Capital C, small c. Capital C I am making like this, okay. Then this is also what? Capital C, small c. So how many of them comes? Let's see that. There will be what? There will be capital A, B, capital C, capital A, capital B, small c, capital A, small b, capital C, capital A, small b, C. Then A, capital B, capital C, A, capital B, small c, A, small capital, A, B, C, small. See how many of them are coming? Let's see that. 1, 2, 3, 4, 5, 6, 7, 8. 8. And what we have, find it over here. See, in a tri-hybrid case, 8 will come. Done. So this is how you have to do it, the Forklin method. Okay. Now my question to you for today is that find the genotype, all possible for the given hybrid. Okay. So first question is X, X, Y, Y, Z, okay, Z, Z, A, A. Okay, you have to write it. And second one is A, A, B, B, C, C, D, D. Okay, or E, E. Okay, so this is your homework for today. Well, I hope that you like this video, and if you like this, give it a like, share it with your friends or your seniors or juniors who might need it for their NEET journey. Moving on to the next thing is that this notes will be available in the WhatsApp group as well as on my Telegram group, which link will be in the comment description box. And well, that's all for this video. Thank you so much.